Welcome to Study I School student, myself Medha Singh, and today I am going to start a lecture series on immunology. So first I explain a basic immunology, the overall process of immunity. So what is immunity? What do you understand by immunity? Term immunity. Immunity is the overall ability of a body, of our body or human body. Immunity is the overall ability of a human body to fight against any foreign pathogen or foreign agent. The foreign agent may be fungi, bacteria or virus. So basically we can divide immunity into two types. The first is innate immunity and the second is acquired immunity. The innate immunity is non-specific kind of immunity and it is also called first line of defense. It is inborn, so that's why it is called innate immunity. And acquired immunity is also called adaptive immunity because we adapt this kind of immunity after birth. So now I am going to explain one by one. Immunity. Innate immunity. Inborn immunity. So we can further classify innate immunity into 4 barriers, and those 4 barriers are cytokine barrier, cellular barrier, physical barrier and physiological barrier. Physical barrier. Number 2 is physiological barrier. Number 3 is cellular barrier. Number 4 is cytokine barrier. So physical barrier, one of the most important physical barrier is our skin. Our skin protect us from many invaders or from many other foreign substances, foreign particles, foreign agent like bacteria, virus, anything else. So skin and mucosa, these two are major kind of physical barrier. Mucosa is generally present in our mucosa is generally present on our urinogenital tract. Our nasal chamber also contains mucosa. Generally bacteria are trapped in this mucosal secretions. And now we further move on physiological barrier. Physiological barrier means physiological barrier generally contains a large number of secretions, body chemical or secretory substances, such as physiological barrier means gastrointestinal juices in our stomach, that is HCl. HCl contains enzymes which kills any foreign substances or bacteria, which generally kills pathogens. Saliva in our mouth and Our tears contain a special type of enzyme which is called lysozyme, which is very important for killing bacteria. Now, cellular barrier. Cellular barrier means all those kind of immune system cells which are present inside our body and fight against all foreign agents or foreign invaders. These cellular barrier contain many type of cells. Such some examples are mostly WBCs. WBCs also called white blood cells of our body or leukocytes. So these contains mainly neutrophils, macrophages, monocytes, etc. Cytokine barrier. Cytokine barrier is mainly interferons. Interferons are generally cytokine barrier. Interferons are mainly active during viral infection. When any viral particle or virus entry inside our body, virus takes entry inside our body, protect us against viral infection. So this is a overall mechanism of basically innate type of immunity. Now we further move an overview of acquired immunity. Acquired immunity is a second line of defense. It is specific. It is pathogen specific. And human can acquire acquired immunity after birth. So that is why it is also called adaptive immunity. Adaptive immunity. So on the basis of working mechanism, this adaptive immunity are basically of two types. The first we can call humoral immunity. Humoral immunity. Humoral immunity and cell-mediated immunity. Cell-mediated immunity. So, what is immunity? Immunity is anything which protect us against any viral particle or any kind of viral, bacterial or fungal particles against pathogens. So what is humoral immunity? The basic difference between humoral immunity and the cell-mediated immunity is that humoral immunity is basically B cell immunity. B lymphocytes are involved in humoral immunity. B lymphocytes involved in humoral immunity. And cell-mediated immunity is basically T cell-mediated immunity. All T lymphocyte, basically T lymphocyte are involved in cell-mediated immunity. T lymphocytes. We all know that B or T lymphocytes are plasma B cells and memory B cells. Plasma B cell stored a memory of first attack when any pathogen enters inside our body for the first time. Then plasma B cells convert into, we can say B cells differentiate into two types of cells. Plasma cells, plasma B cells and Plasma B cells and memory B cells. Plasma B cells converted further into immunoglobulins. Immunoglobulins means antibodies. And memory B cell. Memory B cell, actually memory B cells are specific. When any pathogen enters inside our body for the first time, they create a memory of this first attack. So in near future, when any foreign substances or the same pathogen which attacks for the first time enters second time inside our body, so they can easily activate memory B cells. Contains memory of first attack. I hope students आप लोगों को ये टॉपिक समझ में आ गया होगा. Now you can take a screenshot of this. Now I am going to represent it diagrammatically. What happens when any invader invades our body? First time encounter of any pathogen inside our body. I am going to explain it by diagrammatically. So first I am going to explain innate immunity. Suppose students, this is our body wall. This is our skin. So the first cell which comes out first, that is neutrophil. Neutrophil come out the very first time when any pathogen get entry inside our body. Now working mechanism of neutrophil. Working mechanism of neutrophil. Now working mechanism of neutrophils. You all know that neutrophil are trinucleated cells. We should always represent a neutrophil by 3 nucleus. These nucleoside cells contains lysosome and a phagosome. This is a lysosome and this is a phagosome. All neutrophils kills bacteria by the process of phagocytosis. So we can see a overall view of phagocytosis. Suppose any bacteria get entered inside our body. Just suppose this is a bacteria, or generally just suppose that it is a gram-negative bacteria. So the cell wall of gram-negative bacteria is basically made up of lipopolysaccharide layers. So these are the endotoxin substances of bacteria. Now the bacteria entered for the first time inside our body. Now what will a neutrophil do after that? Firstly a neutrophil catch the pathogen or bacteria, engulf it and cut it down into small pieces. And cut down it into small pieces with the help of different kind of chemical substances or different type of chemical enzymes which is released by lysosomal enzymes. With the help of lysosomal enzymes, the neutrophil engulf any bacteria, cut it up into small pieces, and after that the further procedure will be start. We will see how. Suppose this is the bacteria inside a neutrophil. So what happened after that? Lysosome, this lysosome is fused with the phagosome and make it a phagolysosome. All the lysogenic content which is present inside the lysozyme released inside the bacterial membrane. And at last, lysis of the bacteria occur. Now, after this, all the bacterial content which remains inside the phagolysosome are carried out with the help of different blood cells in the lymphatic vesicle. Lymphatic nodes. Now we will see the mechanism of macrophages. Guys, you all know that macrophages are macro means they are the most important cells of the immune system. Now we are going to discuss about the macrophages. When macrophages present inside our blood vessels, the macrophages is called monocyte, and when it is come outside the blood vessels at the site of infection or inside any tissue, then they convert into macrophages. So remember that when a macrophage is present inside the blood vessels, it is called monocyte, and when it comes outside, then it is called the macrophages. Macrophages are present everywhere. In brain, these cells are called glial cells. In liver, these cells are called Kupffer cells. Macrophage is one of the most important cell in our immune system because they not only kill the pathogen into small fragments, but they cut it down into pieces and showcase it with the help of MHC2. MHC2 means major histocompatibility factor class 2. They showcase the antigen particle with the help of these MHC receptors. With the help of these MHC receptors to rest of the immune system cells. That yes, there is something unwanted substance present inside our body. They call other immune system cells. That all such cells come at the site of infection and helps in killing those foreign particles or those pathogenic material which invades or which present inside our body. So let's move towards macrophages. Suppose this is a gram-negative bacteria which takes entry inside our body. Now macrophages come. This is called nucleus. This is called lysosome. This is called the phagosome. When they fuse together, they make a phagolysosome. And basically this is a receptor. This receptor is called MHC class 2. Major histocompatibility factor class 2. MHC is a special type of protein molecule. When any pathogen enters inside our body, suppose it is a macrophage and it engulfs the bacteria. suppose this is bacteria, and this is phagosome. All the lysosomic content which is present inside the lysosome, all the enzymes released inside this phagolysosome, and after that the lysis of bacteria occur. Now further this antigen molecules, which is showcased with the help of this MHC2 molecule to rest of the immune system cells. This is the antigen. So now a very important, there is a crosstalk between macrophages and T cells are happen. Now this antigen is which is present inside the macrophage, at the top of the macrophage. Now a crosstalk between macrophage and T cells are occur. So here is our T cell. T cell, this is called T0 cell, naive T cell. Naive T cell, when it is activated by chemical signaling through cell communication, and this naive T cell further convert into T helper cell, TH cell. This T helper cell is called CD4 T cells. CD4 T cells. T cells are basically of two types. One is CD4 T cell and the other is CD8 T cells. CD8 T cells are a special kind of T cell. These are called the cytotoxic T cell, which kill all the microbes with the help of special enzymes called granzymes and porphyrins, which create hole inside the wall of bacteria or any kind of pathogen. CD4 is very important in immunity. TH cell, CD4 cells are communicate with macrophages for killing pathogen. Now this TH cell can convert into, after receiving the signal, the cell communication, it converts into CD8 kind of T cell, that is called cytotoxic T cell, or CD4 type means T helper cell. Now after activating T helper cell, now T helper cell starts secreting some interferons, interleukins and cytokine. All these substances are a type of chemical molecule, are a special kind of protein molecule basically, which help in cell communication. And now this T helper cell starts secreting interleukins. The two special kind of interleukin which is secreted by TH cells are interleukin 4 or interleukin 5. IL4 or IL5. Now this IL4 and IL5 is very important for activating other half, that is the immune, that is the adaptive immunity, because these IL4 and IL5 activates B cell, and you all know guys about B cells, that B cells after activating form immunoglobulins or antibody. So B cells finally form antibody. Now with the help of these antibodies, opsonization process occur. All the antibodies, suppose it is a bacteria, all the antibodies kill bacteria finally by the process of opsonization. This is a overall view of immunology. I hope you guys understand this topic. Now you can take a screenshot of that. नाउ आई होप स्टूडेंट्स आप लोगों को ये टॉपिक क्लियर हो गया होगा. ये मैंने अभी डिटेल में नहीं बताया है, इसकी डिटेल्स मैं आप लोगों को अपने फर्दर लेक्चर में दूंगी. ये जस्ट इम्यूनोलॉजी का एक ओवरव्यू था. सो नाउ यू कैन टेक अ स्क्रीनशॉट ऑफ दैट एंड बाय बाय ऑल स्टूडेंट्स.